Okay, so question 1. Figure 1.1 shows a diagram of two sarcomeres of relaxed striated muscle. So you can see that in this drawing, right, there's something that's missing there, okay, which is actually M line, right? There should be an M line here, and there should be another M line here. So it's not there, okay? But overall, the thick and thin is actually correct. So when the striated muscle contracts, state what happens to the length of the I band and A band. So from this question, you know that you need to learn up the structure of these sarcomere very well. So I take opportunity to revise. Okay, can someone tell Jaya Ganesh what is the attendance code? Okay, thank you, Sophia. Right. So let's revise the muscle first. Now, so remember when you learn about muscle, you first learn when you learn about blood vessels, right? There are three layers of tissues, right? So you're starting from endothelium, and then you've got the muscles and the connective tissue. So therefore, muscle, is it an organ or a tissue? Okay, is it an organ? Tissue is a tissue. You can find it everywhere in your body. Now, muscle is a tissue, so inside tissues, there should be muscle cells. So if you look at this structure here, which is a skeletal muscle, right? Inside this tissue, which is called skeletal muscle, there are a lot of muscle cells, which we call them muscle fiber. Because if you remember eating like chicken thigh, right? Having muscles there, or eating some lamb or beef, okay? You can find the muscles have got like fiber-like structure. So each of the fiber is actually a cell. So the cell is called the muscle fiber, okay? Or we call it syncytium, because the cells tend to fuse together, and there are a lot of nucleus. When they fuse together, it seems to have a lot of nucleus. So the cell is called muscle fiber, okay? Muscle is a tissue. The cell is muscle fiber or syncytium. And of course, inside the muscle cells, you will have got the fiber for contraction. So we call them myofibril. Fibril means little fiber. Myo means inside the muscle cell. So myofibril is the little fiber inside the muscle cells. Okay, myofibrils, which is made up of the basic filaments, which is actin and myosin. Okay. So therefore, inside the muscle cell, we have got this myofibril. So each of this circle rod-shaped structure inside the muscle cell is the myofibril, little fiber inside the muscle cells. And inside this myofibril, you've got basically the actin filament and the myosin filament. Okay. So where's my actin and myosin filament? Here, okay, so this one here. So this actin and myosin filament, okay, myosin is the thick one, muscular, and actin is a thin one. So you can see that how they're arranged in such a way that this is how they arrange, and one unit of contraction is called the sarcomere. Okay, one unit of contraction is called the sarcomere. So this is the sarcomere here. So this is one unit of contraction. So it's called a sarcomere. So inside the sarcomere, right in the center, is actually the thick filament. So we call them myosin. Then at the sides are the thin filament, we call them actin. So when the muscle contract, the actin filament, they are supposed to move into the space in between the thick filaments, right? So there are labeling for this structure so that you can actually, you can name them and indicate which part of it you want to mention. So this one you have to memorize, very easy only, okay? The A band is the entire length of the, wait, let me see. So the A band is the entire length of the thick filament. A, because when you draw the two line A, it can overlap. This line and this line can overlap. That means to say that your thin filament can move towards this location one. That's why they can overlap. So it's A band, the entire length of the thick filament. The H band is the region where no overlapping can occur, right? So this space here, where there's no overlapping of thin and thick filament, right? The thick filament region is the H band, right? Then the I band is this region here, where you have your thin filament which do not overlap with the thick filament. So among these three bands, only A band is the one where overlapping can occur between thick and thin filament. Okay, so the entire length. So you must remember. And then for both the thick and thin filament, they must attach to a certain disc. Okay, so therefore you've got the M line or M disc. This is for the thick filament to attach. So it's in the center of the thick filament. Then you've got the Z line or Z disc. This is for the thin filament to attach. Okay, so this one you have to go and learn up, because if it come out in exam, especially with essay question, you better learn up everything. One advice I give to you is please start answering essay question first. Once you get your exam paper 4, please answer essay question first, get all the marks first, and then move on to answer other questions. Okay? And for essay question, remember, you can only choose 1 out of 2 questions. Okay, you cannot say, oh, I'm going to choose question 10A and question 11B. Sorry, I only mark question 10A and B. So you have to choose either 10 or either 11A and B. Okay, so coming back to here, when the muscle contract, okay, when the muscle contracts, so this is what you're going to see here, the changes in the bands, right? So your actin filament will be pulled in between the myosin filament. So what you can see is that your H band will become shorter and shorter. Your I band also becomes shorter and shorter, whereas your A band will never change, because A band is the entire length of the thick filament. It will never change. Only the H and I band become shorter, and hence your entire sarcomere will become shorter and shorter. So this is the entire length of the sarcomere. Can you see it becomes shorter here? Okay. So that's a very basic thing. Please revise them. So answer for this question is the I band becomes shorter and the A band remain the same. Okay. So B, so they say that glycogen storage disease type V. So this is a kind of disease where the glycogen cannot be broken down to release glucose. So when there's no glucose, then there'll be less aerobic respiration, so less quantity of ATP produced. And we know that muscle contractions need ATP. So the question asks the student, with reference to the sliding filament model of muscle contraction, suggest why a lack of ATP affects the functioning of the striated muscle. So lack of ATP, total marks is 3 marks. So that means to say that you need to know what is the function of ATP during muscle contraction. And when it's lack of ATP, whatever stages that need ATP will not function. So you list all of them. So it's very straightforward question. Okay. So going back to the use of ATP in muscle contraction, it starts from here. Okay. So we know that when the muscle is at rest, now muscle is only made of thick filament or thin filament. So go back and revise that your thin filament is made up of 3 components, which is actin, tropomyosin, and troponin. So actin is made up of globular protein that join up together to become an actin chain, like pearl chain. All right, so become filamentous. Then you got tropomyosin, which is a fibrous protein, and troponin. So at rest, our troponin and tropomyosin are blocking the binding site of the myosin head on the actin chain. So it blocks it. So your globular head, the myosin cannot bind. So the thick filament is made up of the myosin head and myosin tail, right? Myosin head is globular, myosin tail is fibrous protein. Okay, so where is ATP needed is here. So step one is, hang on. Now, once action potential arrives to the muscles, then calcium ions will be released from the sarcoplasmic reticulum, and then it will bind to the tropomyosin. So tropomyosin changes shape, and then you see the troponin and tropomyosin move away. See the calcium bind to tropomyosin. So tropomyosin and troponin move away. Am I right? Calcium binds to troponin. Okay, calcium binds to troponin. So troponin changes shape. So tropomyosin and troponin will move together. So it will expose the binding site of myosin head at the actin chain. So then what happens is that myosin head will then be able to bind to the actin chain. All right. And what you can see here is that the ADP, after binding, the ADP has to detach from the myosin head. Then only the head will twist towards the center of the sarcomere, which is the M line, creating the power stroke. So we need the ADP to detach from the myosin head for it to create the power stroke. So the detachment of ADP and PI leads to creation of power stroke. So after the power stroke, we need ATP to bind to the myosin head so that the head can detach from the actin filament. So after detaching, we need the ATP to be hydrolyzed. So you know that once ATP is hydrolyzed, becomes ADP and PI, there's energy released for the head to move back to its original position. So that's the function of ATP. Then after that, then only is able to bind again to the actin. So therefore, based on all the information that is related to ATP, you need to write everything that is related to the ATP. Yeah? So therefore, let's look at the mark scheme answer. Okay, here. So first, the question asks you, what happened when there's lack of ATP? Okay, when there's lack of ATP means no ATP. So then when you don't have ATP, then the myosin head cannot detach from the actin filament. So that's number one. And then because of that, there's also no ATP that can be hydrolyzed to release the energy. So therefore, the head cannot tilt back to its original position or recover. And because of that, there will be no myosin head that can bind to the actin to form a cross bridge. Okay? And then there'll be no ADP that detaches from the myosin head, so there'll be no power stroke. And also, calcium ions having released from the sarcoplasmic reticulum, it has to be pumped back to be stored in the sarcoplasmic reticulum. So when there's no ATP, there's no active transport of calcium ions back to sarcoplasmic reticulum. Okay, so that means you need to be familiar with all the steps very, very well for this one. So it's very easy. Okay, if you study, you can answer question. You don't study well, you cannot answer question. Okay, so please revise them all.